Today we are going to learn how to make a peripheral smear and determination of DLC. Apparatus required for the practical are 4 to 5 clean glass slides, Leishman stain, glass dropper, cotton swab, distilled water, pricking needle or lancet in case of finger prick method, but here we'll be using EDTA vial blood, and a compound microscope. Starting with the procedure of making peripheral smear, take 4 to 5 clean and grease-free slides with smooth edges. Pour a drop of blood in the center about 1 to 2 cm from one end of the slide. Place the edge of the spreader on the surface of slide just in front of a blood drop at an angle of 45 degrees. Draw the spreader backward so as to touch the blood drop and hold there till the blood run along the full width of the spreader at the line of junction. The spreader is then moved slowly and smoothly to the other end of the slide, maintaining the same angle. Allow the smear to dry and air. The peripheral smear has 3 parts: head, body and tail. Area between body and tail is ideal for the cell count. Now moving on to the fixing and the staining procedure of the peripheral smear. Place the blood smear across 2 parallel support on the rod. Pour 8 to 10 drops of Leishman stain on each slide, just enough to cover the blood smear. Leave it for 2 minutes. This is called as fixation time. At the end of 2 minutes, add equal number of drops of distilled water over the stain. Mix the stain and blood evenly by gentle mixing with the dropper. Leave the dilution for 10 minutes, and this 10 minutes is the time of staining. After the completion of staining time, at the end of 10 minutes, pour off the stain and hold the slide in slanting position, and water is allowed to float over the smear. Let the smear dry completely, and after that focus it under the microscope. At 100x under oil immersion, the cell count is done. A minimum of 100 WBCs are identified in a systematic manner, and counting is made using the telebar method. Ideally, all the cells should be counted in a single strip running the length of the smear, proceeding from the base to the apex. Use the mechanical stage to traverse the full length of the smear. Move the slide along vertically by a distance equivalent to 2 mm and again traverse the full length of the smear, this time in opposite direction. This method ensures that cells are counted not more than once. For the calculation of DLC, 100 WBCs are counted and noted in the square like this. Here are few pictures of different WBCs focused under the microscope.